Hi everyone, my name is Tang Li. My poster presentation today will introduce our recent study, Critical Assessment of Pan-genomics of Metagenome-Assembled Genomes. First, I will give a brief introduction for pan-genome. Pan-genome represents the entire gene site of all strains in a species. Homologous genes will be clustered based on their sequence identity. Core genes are shared by all strains, while dispensable genes present in some of the strains. Unique genes are only found in specific strains. And what is metagenome-assembled genomes? Metagenome-assembled genomes are produced from metagenome sequencing rates through filtering, assembly, binning, and taxonomic assignments to generate the approximate representation of actual individual genomes. Due to the computational process, the nature of MACs are fragmentation, incompleteness, and contamination. Therefore, pan-genome analysis of MACs may suffer from these known issues. In recent years, pan-genomics of MACs are increasingly used in microbiome research. Three pan-genome analysis tools, ANVIL, RORI, and BPG, are the most used. Here we conduct a critical assessment of pan-genomics of MACs by comparing pan-genome analysis results of complete bacterial genomes and simulated MACs by using these three tools. In our simulation method, complete bacterial genomes were randomly cut into fragments. Then a part of the sequence was randomly removed from each fragment to create gaps. After that, contaminations were added by inserting sequences from closely related species. Since MACs are often combined with complete isolated genomes together for pan-genome analysis, mixed datasets were created by randomly selecting MACs from the simulated MAC dataset and combining with the complete genomes. We used 17 bacterial species in our study. Here I just use Escherichia coli as example species to show our results. First, we found that core genome size dramatically decreased with genome fragmentation and incompleteness in all three tools. However, the number of core genes increased slightly when using BPG and ANVIL in the contamination datasets, while the opposite results in RORI might caused by the possible bias in the gene clustering step. The core gene loss remains when using mixed datasets. We found that the higher percentage of MACs in the datasets, the more core gene will be lost. The strictest core gene threshold is 100%. However, in practice, core gene can be defined with a more relaxed threshold like 95%. The lower core gene thresholds always give a higher core genome percentage and also the larger genome size, meaning that core gene loss can be alleviated by the lower core gene thresholds. What's more, the ANVIL with its default metagenome modes for gene prediction performs better than the other tools in fragmented genomes, shown in these plots. In pan-genome analysis, after the core and accessory genes are identified, downstream analysis are often performed on these genes to understand the genome evolution and the gene functions. We performed the cluster ortholog gene analysis to find enriched functions in core genome. When using the 100% core gene threshold, the number of core gene families assigned to core categories was dramatically decreased with increasing fragmentation and incompleteness. For the phylogenetic analysis, the phylogenetic trees can be built based on the single copy core gene alignment or the gene presence and absence matrix. By quantifying the topology changed between the trees before and after simulation, the results indicate that the more fragmentation, incompleteness, and contamination MAC have, the less accurate phylogeny will be in respective of tree construction methods. Lastly, we show the process of core gene loss with fragmentation, incompleteness, and contamination. If fragmentation disrupts a gene coding region, the gene will be lost due to missing of start or stop codons. More gene might be partially or fully removed due to the deletion caused by the incompleteness step. Contamination from closely related strains or species should not lead to core gene loss due to no removal of existing genomic regions. All in all, incompleteness of MACs lead to more significant core gene loss than fragmentation and contamination. Core gene loss remains when using different pan-genomic tools and when using a mixture of MACs and complete genomes. In the pan-genome analysis, using lower core gene threshold and predicting genes in metagenome modes are necessary to elevate core gene loss. It also helps to improve gene functional enrichment analysis and phylogenetic analysis. Combining the complete isolated genome with MACs for pan-genome analysis is better than just use MAC datasets. Lastly, new pan-genome analysis tools considering the characteristic of MACs should be especially designed in the future studies. Thank you for listening, and please feel free to make any comments if you have any questions.